Now that we hopefully understand a little bit more all these physics things that play around with us, we're going to talk about what we put in our samples. So fluorescence and fluorophores. I think remove all these things that keep popping up. So fluorescence is a cycle of photon absorption and photon emission. So only, to start with, only some molecules can fluoresce, not all of them, only some of them, right? And they are called fluorophores or fluorochrome, it's the same thing. When we excite a fluorophore, it acts like a light source in the sample. So we have these lots of tiny light sources in our sample. So you can see in here, maybe I can do that. Yes, we can see in here that we're shining some light at a certain wavelength. And here it's UV light, so it's high energy light on our molecule, which is a fluorophore. And then the electrons around the core of the molecule will get excited. And so they go into an excited state. And with this, they get energy. And when they get this energy, they vibrate, and then they lose heat by vibrating like this. But anyway, they do not lose all the heat, maybe. And so what they do is spit out the heat that they have in form of light. So this is the emitted light. And you can see that the light that is coming in has a different color compared to the light coming out. And that is because of all this energy that has been lost by vibration at the time when the fluorophore was excited. So we have an absorption of light, we have a loss of light through vibration, loss of energy through vibration, and then we have emission of energy. And this is the reason why we have two different lights when we deal with fluorescence. There's always two lights. There's the excitation light, and then there is the emission light, and there is a shift between them. And the emission light, in general, for almost all the case, is shifted toward longer wavelengths. So longer wavelengths, if you remember, is lower in energy. And that is because we have a loss of energy in here. So the excitation light has more energy than the emission light. So the emission light is always shifted towards the red side of the spectrum. This is true always, except for multiphoton excitation, which we will not talk about in this course, but in that case, the excitation light is longer than the emission light. So this is often shown as this energy diagram called the Jablonski energy diagram, which shows everything that can happen when we excite the fluorophore. So here we are at what is called the ground state. So this is the fluorophore being in the sample and not being excited at all. There's no light, for example. Then we shine the excitation light on this sample. And what happens, the fluorophore gets excited, and boom, it shoots up into this excited state, which is called the singlet state. Okay, and then it vibrates because too much energy, and so it's a loss of energy and a lot through heat, and the fluorophore goes into a different singlet state, which has a lower energy. It's very unstable. All these things are very unstable, and so there are different options. When the fluorophore is excited, several things can happen. One of them is that the fluorophore shines lights and poof, it goes back to the ground state. Okay, so this is emission. This is fluorescence emission. But some other stuff can happen then. What can happen is that simply the fluorophore keeps on losing, like shaking and losing energy, and it does not fluoresce. That's possible. But another thing that can happen is that it actually goes into another state called the triplet state. And in the triplet state, you see it's delayed fluorescence, which means that it stays in this state for a little while, and this is actually quite stable. And then after a while, it fluoresces, or it goes back through shaking, losing energy. Okay, so this process of fluorescing and emitting light, if the fluorophore, when it's excited, goes into the triplet state, then the fluorescence is delayed. And we will see later what implication all this has for us. So the fluorophores can be in several states. The relaxed state is very stable, it's the ground state, but the singlet state, the excited singlet state, is very unstable. And the dark state, this is also called the dark state or triplet state, it's the same thing, is actually the fluorophore is excited, but it's stable there in that state and it does not fluoresce until later it does fluoresce. Okay, so when the fluorophore is here, it is not fluorescent, it doesn't shine any energy. It just stays there and that is stable. So when we talk about samples emitting light, we have different words, and I thought that maybe I could clarify what it is. Luminescence is all phenomena where light is emitted. So fluorescence is one type of luminescence. This means luminescence means there's production of light. Chemiluminescence is luminescence that is not triggered by light excitation, but by chemical reaction. So we have a chemical reaction and then the compound will shine light. So this is chemiluminescence. Bioluminescence is just natural luminescence. So it's actually animals and plants and corals and stuff that shine light is called bioluminescence. And phosphorescence, fluorescence are actually luminescence triggered by light. So it's all triggered some sort of luminescence that we trigger by light. Both of them are the same phenomenon, but this one, it goes slower because we have a transition through the dark state. In these guys go to the dark state, and so they take a while before they actually fluoresce. And this is the reason why we have a difference in time between fluorescence and phosphorescence, but it's the same type of phenomenon. So going into the dark state, or also called triplet state, is a statistical process. So sometimes the fluorophore, when it's excited, will fluoresce. Sometimes it will not fluoresce, it will just vibrate. Sometimes it will go into the dark state. If you shine very, very strong light on your fluorophore, there is a higher risk to send the fluorophores to the dark state, especially if you shine very strong light for a long time. So if you force the fluorophores to do lots of cycles like this, at some point, it's a statistical thing, it will end up in the dark state and it will stay there and it will not fluoresce. When we image with high illumination power, push the laser very high, we would like to image for a very short time. If we combine imaging with very high power and imaging for a long time, then we will send our fluorophores to the dark state, and you will see why we don't want to do that. So this has consequences on our settings. For example, if you use a single point confocal, you would want to use some averaging, some frame averaging, instead of slowing down the speed. You know, on the point confocal, you have something called the speed, or it's also called the pixel dwell time. So this is how long we stay on one pixel before we move to the next pixel. And so that means we shine very strong light for a very long time on one pixel, and what we do is that we actually effectively send our fluorophores to the dark state. Something else can happen when the fluorophore is up there in the excited state. It is photobleaching and photo damage. So when the fluorophores are up there in the excited state, if they encounter oxygen, if they interact with oxygen, there is an irreversible destruction of the fluorophore. So irreversible, this is bleaching, and it cannot go back. And also it produces reactive oxygen species that this destruction of the fluorophore produces this reactive oxygen species, and that leads to photo damage. So this is a destruction or a modification of the macromolecule inside our cells. Clearly, this is not something that we want. So when we image live samples, this is what happens. We have a sample that can be damaged and react to the damage. When it's fixed, it can be damaged but not react to the damage because it's fixed. But if we have a live sample, we have to be very careful with photo damage, and this is exactly what happens. It means the fluorophores are in the excited state and there is oxygen coming in. So how can we prevent oxygen, the oxidation by oxygen in the medium? There are several ways. We can put some chemicals which are oxygen scavengers, and this is what we have in our anti-fade reagents. So when we have a mounting medium, there is always anti-fade reagents in that, and what they do is that they prevent the excited fluorophores from being destroyed by oxygen, by scavenging the oxygen, okay? So this is why mounting medium are very good, because they preserve your sample chemically. But we can also physically help by putting a cover slip on the sample, and if the cover slip touches the medium. So even if we have a live sample, if we want to prevent bleaching and photo damage, we can actually seal the medium with a cover slip, and that helps in preventing this photo damage. But clearly, on the long term, it doesn't work. But for a short term, it works. This is something that people use in a storm, for example, if they image live. So this is how bleaching and photo damage work. And what you can see in here is that bleaching is not linear at all. So if it starts very, very quickly and then it slows down. So this is not a linear process. So you do not want to look at the fluorescence in the eyepiece if you can avoid this. And I know that this is common, that people do that. They look at the fluorescence in the eyepiece, and what do? They try to focus on their sample with DAPI. This is not a good practice. As you know, DAPI uses a short wavelength to be excited. So you're shining some short wavelengths with very high energy onto your sample, and you shine it for a long time because you're there. Which cell am I going to image? Oh, nice. Oh, I'm not completely in focus. I'm going a little bit more, a little bit less. So you take a long time with some short wavelengths, so high energy light. And what you're doing is that you're going into this and you're bleaching your sample. And one of the main issues is that you may not bleach DAPI, but you may bleach the green as well, because you can look at the spectra when you're imaging. The green fluorophores are very often excited also at the wavelengths that we use for DAPI. Okay, so avoid looking at fluorescence in the eyepiece, and if you have no other choice, you should do this as little as possible. And we will talk later about how we can use the automation of the microscope to avoid having to look at the fluorescence in the eyepiece. So now I'm going to talk about the different types of fluorophores. So there are basically two types of fluorophores. And maybe those people from Ireland here who are developing fluorophores, if I say something wrong, I want you to tell me. Thank you. So there are some fluorophores that are produced by chemistry, so chemical fluorophores, and there are fluorophores that will be produced by the cells, and these are protein fluorophores, okay? So, and of course, we have also autofluorescence, and that can be just anything. It can be, so it's inside the cells, inside the tissue. It could be due to proteins that fluoresce, micromolecules that fluoresce, metabolites, pigments, anything you want. So autofluorescence is also there. But the fluorophores that we are using are either chemical fluorophores or protein fluorophores. So let's have a look a little bit at chemical fluorophores. We have dyes, all these organic dyes they use. So I'm sure that you recognize all these names, all the Alexa fluorophores and FITs and TRITs and stuff like this are organic dyes. So they are chemical fluorophores. And they can be really specialized to mark DNA, membrane, cytoplasm, mitochondria. You can use these chemical fluorophores in live cells or in fixed cells. So you recognize certainly some of the names in here. And so these are some types of fluorophores. We also have specialized fluorophores, like those that change wavelengths or change intensity based on something around. This is what you use, for example, to measure pH or calcium concentration. So in this, you can see a neurosphere with flip calcium 5 that measures the calcium pulses, and the intensity increases when there is calcium. So this is one chemical fluorophore that has that works this way. Now, another type of chemical fluorophore that has been very useful since the advent of super resolution is blinking fluorophores. These fluorophores are prone to going to the dark state, so they have a higher statistical chance to, when they're excited, to go into the dark state and wait there. And then later they fluoresce. And this is why it's blinking, because there's a delay between the excitation and the emission. So these are, for example, these fluorophores, and this is super useful for super resolution like Storm and Palm. So these are specialized. So now I was telling you about the chemical fluorophore, but now I'm going to talk about the protein fluorophores. So these proteins, we have a tendency, we like to fuse them with our protein of interest. So you know a lot of them, EGFP is one of them. And we will make a plasmid with the cDNA, then we transfect and fuse to the protein of interest, and then we transfect the cells, and it's the cells that produces these proteins. With their own machinery. So then we can look at the cells live or fixed. But that's one of the main disadvantages of this is that it requires overexpression of a highly modified protein. So we need to make sure that this does not interfere with what we want to measure. And the artifacts are very common. So it's estimated to, and I have the paper in here, if you want to look, estimated to around 30% of all fusion proteins will create some sort of artifact. But unfortunately in biology, we don't really check. So we should definitely check more when we make a new fusion protein that has not been made before. We should check that everything works as it should in the cell, and that there is no difference with when we do not overexpress protein. So there can be lots of different artifacts like the maturation, localization, maybe the protein goes to the wrong place, it interacts with something, etc. So unfortunately, I mean, these protein fluorophores are very, very useful, but unfortunately we are not doing our job by doing other controls when we produce a new one. So the fluorescent proteins come in many colors, the dyes as well. Lots of different colors. They were basically started being evolved from jellyfish or also corals, and that gives a very enormous tree of different types of fluorescent proteins. And over the years, more and more have been evolved that we can excite at different wavelengths, that fluoresce at different wavelengths. And so they are new and better fluorophores that are created every single year. So I really encourage you to question the fluorophore, the protein fluorophore that you are using, and I will show you how to check for which is the best fluorescent protein, the latest one that you could use, and you can easily double up the fluorescence that you have, or even a lot more if you just change protein. So that can be a great advantage. So there are also fluorescent proteins that are specialized. For example, some of them are photo switchable. So they can start with one color, and then when you activate them, they turn to a different color. This is the case of MK8A, for example, that goes from green to red. And we also have some photoactivatable that are either on or off. So again, it's off. And then when you do a special treatment with light, then it turns on. So it's quite useful. Here, this is kaede that turns from green to red in a transgenic fish. And what you can see in here is, is that we have the head of the fish. And here we're shining some light that turns kaede from green to red. So we see all the rest that has not been activated to become red, but it means that these cells that have been photoactivated to turn red, we can then follow them in a live zebrafish and see where they go, and have as a control the cells that were not photoactivated. So this is some specialized fluorescent proteins. Another type of fluorescent proteins are specialized as those that are used for bimolecular fluorescence complementation, or BFC, where we have actually on we fuse one molecule of interest onto half of the fluorophore and the other molecule of interest onto the other half. And when these two molecules come together, then the fluorophore is reconstructed and it starts fluorescing. So that is one type of ways to see molecular interaction. Another way to see molecular interaction is FRET, which is called Förster resonance energy transfer. And what happens here, we can have two proteins, one labeled with a fluorophore, for example, YFP, and another protein of interest labeled with another fluorophore, like, for example, CFP. And with the two proteins interact with each other, so they come very close to each other. The two fluorophores get close to each other, and there is energy transfer between one to the other. And this one will start fluorescing even though we do not excite it. So this is an example of cells that are transfected with a FRET biosensor, which shows histone methylation. So when histones are methylated, they change conformation and the two fluorophores come close to each other. And so what you see in this is that we will have different colors in the cells. Red means a high methylation, blue means a low methylation, and green means intermediate, because it's color coded. So you can see the cells are there and they're all blue, 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 blue. And then at some point the cells start dividing and then it turns red. Yeah. So whenever the cells divide, we have methylation of the histones, and we can visualize this because of the FRET biosensor, which means that the biosensor starts fluorescing. So this is another example of a biosensor where we have a tension sensor. So these are the focal adhesions. I'm sure that someone, I can't remember who was working on focal adhesions, will be interested in there. So in there, we have all the focal adhesions at the edge of these live cells. And what happens is that when there is a change in the conformation of integrin, I think it was there, then the color changes, basically. So this is also a biosensor. So one more thing that I want to mention about fluorescence or fluorophores is FLIM. So we have now this year quite a few people who work with FLIM. FLIM stands for Fluorescence Lifetime Imaging. When we talk about fluorophores, we often talk about the excitation and the emission light, and this is what we are very used to, which excitation it has with emission. But actually, every fluorophore also have a lifetime, and this lifetime is measured in nanoseconds. So what is the lifetime? It means if I have one fluorophore and I shine some excitation light in a very, very short pulse, so bloop, just a bit of light and then turn it off, the fluorophores will get excited and then they will start emitting light. And then they will emit light for a certain time. And then they will stop emitting. So they get all this energy from the excitation light, they vibrate, and then they start sending photons. But then at some point they go back to the ground state and they stop emitting. So how long does this process of emission after a very short pulse of excitation, how long does it last? And what is the shape of the curve? This is what the lifetime is. So the lifetime of the fluorophore is this curve in here, and there's a way to calculate just the lifetime, and it's in nanoseconds. Okay, for how many nanoseconds the fluorophore is going to shine to emit some light after a very short burst of excitation. So this lifetime is extremely useful because it changes with the environment of the fluorophore. So if the pH is different, we're going to have a different lifetime. And if the molecule interacts with other molecules, then we may have a different lifetime as well. So this is really useful because it means that, for example, you can visualize the same molecule with a different lifetime in different environment. If it's inside an endosome or if it's outside the endosome, you're going to get a different lifetime, even though it's the same molecule. The excitation and the emission spectra do not change, but the lifetime will change based on the environment. So here I have a little movie as well, where you can see some cells that are moving, and then I think, I can't remember, there is something that happens, or a pulse of something, and then you can see that some of the cells will suddenly be activated, and all this, the lifetime is changing. So you can see that it goes to 2.8 nanosecond, whereas before it was 2.2 nanoseconds. So there's a very small difference, but it is very reliable, and it means that you can measure, and then slowly it goes back. The cells go back to the state where they were before. So as we can differentiate fluorophores based on their excitation and emission, we can also differentiate them on their lifetime. Now I have spoken about the different types of fluorophores, chemicals or proteins, and there are also some hybrid techniques that combine protein with a chemical dye. For example, the modification of the protein with a chemical dye. And this is things that you have heard probably: click chemistry, SnapTAC, we have flash rehash, genetic code expansion. All these techniques actually combine the overexpression of a protein with a chemical dye. So we overexpress the protein of interest. So we're not fusing the protein of interest with a fluorophore. We're just modifying, we do a tiny modification of the protein of interest. So for example, we can just change one amino acid in the protein, and we overexpress this abnormal construct into the cell. But this protein is not fused to fluorophore. But because of the tiny modification, we can now add a labeled reagent, which is cell permanent, is going to go into the cell and it's going to bind very strongly to this little modification on the protein, and that stuff is labeled. So this is extremely specific, and it has less disruption of the conformation of the protein. You can imagine if we have a small protein of interest and we attach a very big fluorophore like GFP or something, it's not GFP, not super big, a big fluorophore to the protein of interest, sometimes we can create more artifacts than in here. We have a very small protein, so modification in the protein, so we have less artifacts in this case. So lower disruption of the protein conformation. But unfortunately, it also requires overexpression of the modified protein. So we anyway may have some artifacts due to overexpression. So these were the hybrid techniques. So how can you pick the best fluorescent protein for your experiments? I told you yesterday that, yes, when you go to a lab, you inherit lots of things, the whole history and all the good stuff and also all the bad stuff, unfortunately. So your lab may not be expert in fluorescence, and they might not realize that the fluorophore that they have always used, use that one. This is the one the whole lab has been using forever. Yeah, great. But the fluorophores evolve and they get better and better. And so you may, just by changing the fluorescent protein that you're using, you may really improve a lot your results. And you may help your lab by telling them, okay, let's change our construct. So there's a fantastic tool for how to pick the best fluorescent protein. It's called FP-Base, FP for fluorescent protein, base.org. And it's very, very good. And if you will have an exercise where you will have to actually find some stuff on FP-Base, so you will know how to use it. But this is a screenshot of the interactive chart and the advanced search. And you can, select the excitation of that you want. If you're looking for a good fluorophore, you can select all sorts of stuff and display it in a different way. And then you will see the excitation and emission spectra that for example, you can find the fluorescent protein that best match your microscope light source and filter. By doing this, you can select in here. You can also find the fluorescence lifetime. So if you are imaging with FLIM, and how long the fluorophore emits after being excited. There's also maturation. So you can see in here all the stuff that are for each fluorophore. The maturation time is the fluorescent protein. So basically the maturation time is how long does it take for the fluorescent protein to fold and be ready to, so when it's produced by the cell, because the protein is produced by the cell, how long does it take to mature and fold so that it starts fluorescing? If you have a protein of interest that folds very quickly and your fluorescent tag folds very slowly, it's going to slow down the whole protein, and that's not very good. So you want to also pay attention to the maturation time when you choose the fluorescent protein of your choice. And then another thing that you can see here is the photostability. So stability is how fast the fluorophore bleaches. Not all fluorophores are equal in the face of bleaching. So the coefficient, this one, EC, is the extension coefficient. That one is the capacity to absorb light. Some proteins just don't absorb light very well. So you want something where you shine light on your sample that the fluorophores absorb that light, right? And then the quantum yield, which is the QY here, is the number of photons that are emitted So every time the fluorescent protein absorbs a photon, how many photons does it emit? We want to have one photon in, one photon out, but sometimes it's not the case. And so we would like to have a quantum yield of 1 if we want an excellent fluorescent protein. And then, so the brightness of the protein is actually these two multiplied by each other. So brightness is very, very useful as well to see, okay, of course you want to have a bright fluorescent protein, right? So then there is something else that is not in here, but it's actually below the chart. I didn't put it in my screenshot, and it's the oligomerization. And that is very important. Your fluorescent protein that you're going to tag your protein of interest with should be a monomer. We do not want our fusion protein with the protein of interest and the fluorescence protein. We don't want them to dimerize. If the protein of interest should not dimerize, you really don't want this to dimerize because the fluorescent tag is dimerizing, right? So we really want to have a monomer as a fluorescent tag. And so this is basically below there, you will see, but you will get to practice that. So even a weak dimerization is a problem if your proteins are concentrated. For example, if you have a membrane or vesicle protein of interest and you fuse it to a protein fluorophore that dimerizes, even if it dimerizes weakly, because of the high concentrations, there is a risk that it dimerizes, and then the protein of interest behaves in a different way. Okay, so the oligomerization is important. So you're going to have to check all this, and you will practice that. So how do we pick then the best chemical dye? Because the FP base is only for fluorescent proteins. How do we pick the best? So there is, sorry, I changed this. There are information here for the for chemical dyes on FP base, but you don't have this very nice interactive chart, unfortunately. So how to pick the best chemical dye? So again, if you change the fluorophore that your lab has always been using, you may dramatically improve your results. So it's the same thing exactly that we have the excitation and emission spectra, we have a lifetime, but unfortunately, so we don't have the maturation lifetime because this is only for proteins. We also have a photostability, some dyes bleach faster than others, and we also have the brightness, which is these two together. So different dyes will be brighter or dimmer. And we don't have the problem of oligomerization because this is only for proteins. So unfortunately, there is no nice database like FP base, but we have the molecular handbook. The molecular probe handbook has a lot of information about dyes, and this is kept up to date by Fisher, I think. It's part of, Thermo Fisher. So you can get this, and it's very nice. So now we have seen fluorescent proteins and fluorescent dyes. And for dyes, I really encourage you to write to the company and ask them if you have a dye and you think it's not super optimal, you would like to know if there's something brighter. Write to the company that sells it and then ask, do you have anything brighter than this? And they will tell you. So how do we deal with autofluorescence then? So we will see this a little bit more in the sample preparation module. But basically, we can do spectral unmixing. Some chemical treatments are possible as well. There's bleaching possible. And one thing that people do not think about, just think about getting rid of the autofluorescence, but autofluorescence can be a great advantage where you do not need to label something to see it. So in this case that I have here, this is a heart section. Human heart section is highly autofluorescent, and you see after the chemical treatment, all this green autofluorescence that was everywhere is gone. This is Sudan black. So autofluorescence can be a great advantage, and I think we often see in the samples from the students in here. So here is a conclusion for this lecture. So we have, please choose bright and photostable and monomeric fluorescent proteins in FP base. You should check for efficient excitation and emission on that it works for the microscope you use, and pick fluorophores that have a low risk for bleed through on the microscope you use. So this is the stuff that we're going to do this afternoon, this workshop that we will do this afternoon. And also ask the company for the best chemical fluorophores for your application. They will be more than happy if they can sell you a new fluorophore. They will be more than happy to give you some advice. So please do not pick a fluorophore from the fridge just before because it's there. Be critical and try to evolve the things that you have in your lab. Right, so now I'm going to stop sharing and I want to know if there are any questions.
So I have a quick question. Maybe there isn't an easy answer for this. Are there any guidance on how different fluorophores react in different chemical environments? So, for example, when you do clearing and you have different pHs and so on, it's quite common for fluorophores to work or not. And usually, what I've been doing is just looking if the author of the paper has tested the exact fluorophore. Are there any like general guidance that a certain type of fluorophore works in a certain pH or such resources?
Yeah, I am not aware of. I think there are some papers where, with extensive testing of a lot of different fluorophores with different clearing, what happens in proteins, some proteins need water to fluoresce. For example, EGFP is one of them. So you need it like a barrel, you need to have water inside for the molecule to fluoresce. And so when some clearing techniques dehydrate the sample. And when you do that, you remove the water so the fluorophore stops fluorescing. So it's very annoying. If you have a EGFP, your mouse expresses EGFP, and then you dehydrate no more EGFP, then you have to do an antibody staining against GFP to get your fluorescence back. But some fluorescent proteins are more prone to doing this than others. And it's the same thing for dyes. So I think it's a case of matching the fluorophore with the technique. So there are so many clearing techniques that, it's quite often there's a new protocol that comes up that just skips one step of the previous protocol. And so maybe so there's a lot of work on trying to get protocols that clear very well and preserve fluorescence. So I think for that, there's no rule really. I think checking the latest protocol as you're doing is likely the best option.
Thank you.
I also had a quick question.
Yes.
Is there any database or way to get information about how toxic different fluorescent proteins are? I know, for example, GFP, if too highly expressed, can be toxic for the cell or for certain cells. Is there anything like that?
So this is actually, unfortunately, the case for all fluorescent proteins. I'm not aware of any database that will say if it's more toxic or not toxic. The problem of the toxicity, I think, comes a lot from the photostability of the protein. Because if you're working with live samples and you don't want all these reactive oxygens and stuff like this, and some of the fluorophores are more prone to bleaching, and you can have more reactive oxygen species flying around and destroying your sample. But also, I think that in general, overexpressing a protein is a problem in a cell. So, this is not a natural situation. We're making the cell like a big bag of something that is highly modified. And when we get too much expression in the cells, then it's very common that the fluorescent protein gets stuck in the ER, for example, and you're going to have some ER staining that should not be there. And clearly that affects the cells. So I think I'm not aware of any specific proteins that are not good and toxic, but I'm aware that too much overexpression is the problem. So I might be wrong, but I think this is the way it is. So it's very important to overexpress the protein as little as possible. If you make a new cell line, do not pick the brightest ones. If you want to make a stable cell line and you transfect, don't pick the brightest ones, because they are very, very likely to be totally screwed up.
Okay, thank you.
You guys have any more questions?